Good, sir.
Good. Okay, great. So, um, yeah, let's start with today's class. Can you see the screen that I'm sharing?
Yes,
Notebook.
sir.
Perfect. Okay, very well then. Um, you were present in the last class, right?
No, sir.
No, you were not present. Uh, so I think you missed quite some bit then, and I'm not sure how. Let me give you a quick summary of what we did in the last class, and then that maybe can help you, come to the page from where we'll start today. So, were you present in the class before the last class?
Yes,
Okay, great. So you know that we talked about the plasmids, right?
yes,
Which we can use as cloning vectors. We can also use viruses uh, after making some modifications, also as vectors. So in the last class, we talked about what basically are the characteristic features that are required in every cloning vector. Every cloning vector should have those features. The first feature was origin of replication. And if you don't know what is origin of replication, it's simply a sequence that allows the DNA to replicate inside a cell. So as we know, even the chromosomes that we have, when they replicate, it's not like they randomly can replicate replication. And that particular specific sequence is called origin of replication sequence. Okay. So
yes, sir.
every cloning vector should also have a origin of replication. And can you tell me why is that important? What if a vector does not have origin of replication? Why are we using a vector for? What are we using a vector for? Aisha? Just try. It's not a test that I'm taking. Okay. We are two colleagues discussing science and logic. So think about it. Why are we what are we using a cloning vector for?
To transfer the DNA.
Perfect. So you know the answers. Why you people don't speak? Are you being punished at school for giving wrong answers? Is that why you have the inherent hesitance against giving wrong answers? You should give answers, without any fear. Even if it's wrong, it's wrong. What you going to lose? Nothing. You gave a wrong answer, you will get to know what was the right answer. From next time onwards, you will give a right answer. Simple, right? And
Yes,
you
sir.
know the right answer. In this case, you are right. We need a cloning vector because we want to put our desired DNA in it and want to send it inside a host cell where it will do what? Why are we doing this? So that the host cell can make multiple copies of that gene for us, can transcribe and translate that gene for us, and give us the final protein product, right? This is why we are doing all this, correct?
Yes, sir.
Yes. So that's why origin of replication is very important, because without that, that simple piece of DNA which we have inserted inside the host will not replicate on its own. Okay. So our plasmids need to have origin of replication. Second important feature that it should have is a selectable marker gene. Now, this selectable marker is used to identify transformants cells. Transformants are those cells which have successfully taken our DNA and we would like to identify them, by some method. So most of the time, these selectable marker genes are antibiotic resistant genes. Okay. Third is the cloning site, and cloning site is nothing but simply the region where the endo restriction endonuclease recognize and cleave the DNA, cut the DNA, right? Okay, we
Yeah.
have Parker also joining in. Good evening, Parker. How are you doing? Can you hear me, Parker? Because I can't hear you. You're speaking. Okay. See if your mic is functioning, because couldn't hear you. And then I'm just giving a quick recap of last class, and then we'll start with today's class. And in the last class, then we talked about this standard cloning vector known as the PBR 322. And all these sites that you see here are the restriction endonuclease sites, BAMH1, SAL1. These are the sites where these respective restriction endonuclease can cut the plasmid, and these are called cloning sites or recognition sites. Okay, good evening, Iram and Rabia.
Good evening.
Yeah. And then we talked about another method of insertional activation where we do not have to any longer use an antibiotic resistant gene as a selectable marker. There are some pros, but some cons of using antibiotic resistant genes because they are difficult to manage. You have to make multiple cultures. Plus, you are for no, like unnecessarily, you are exposing a lot of bacteria to antibiotic, and there are more chances to develop resistance because we are providing this antibiotic resistant gene. Okay, so instead we can use another method where we use the gene for beta galactosidase instead of Parker's mic is not working. Okay, I think it's some issue with your setting, maybe Parker, the Zoom setting, if it happened all of a sudden in the previous class. Just try checking that while you are. Yeah, we can hear some static when you are unmuting yourself, some static noise, but not a clear voice. Maybe just try plugging in if it's a audio device that you are using or a mic you are using. Or try using a Bluetooth or something, maybe that can help. Okay, so coming back, beta galactosidase is a gene for an enzyme that can react with some substrate, and when it reacts, it gives blue colonies. So if you have beta galactosidase gene active, you will get colonies growing as blue in color. Okay. And simply, if it's active, it simply means that you have not cut it and not inserted your gene of interest. So beta galactosidase active means your gene is not present. So the bacteria are non-transformants. Okay. Beta galactosidase gene has been cleaved and something has been inserted. Whatever you wanted to insert have been inserted successfully, and hence the colonies are colorless and those are transformants. This is a very easy way to identify using the selectable marker through this approach. Okay. At the end, we talked about, we asked a question. All of this is okay. Having said all of this is good, but why will any cell be willing to take up a foreign piece of DNA? And the answer is mostly they won't. So what we can do, we have to prepare them so that they can take up the DNA. Now there are multiple methods to do that. One of the method which we discussed in the last class was a heat shock method using a divalent cation. Now, what was the heat shock method? In the heat shock method, what we do is we keep our cells, whatever cells we want to make transformant, competent, we keep them in ice basically for a long time. We incubate them with the recombinant DNA on ice. So there has to be your DNA, which is the plasmid that you want to go inside. and they are on ice. So nothing will happen till they are on ice. Then for a very brief moment, like few seconds, you will put it on 42 degrees. Now coming from ice to 42 degree, that is a big change. And that gives kind of the heat shock to these cells. And due to this heat shock, the small pores which are present in the bacterial membrane and the cell wall, they kind of momentarily enlarge and then come back to their original size when you put it back on ice. So it's like incubate on ice, briefly for seconds on 42 degree, and then put back on ice. This is a way by which you can make bacterial cells competent and they can take up the foreign DNA. So far, so good. Clear to everyone? Yes, no? Anyone has any doubt?
Mhm.
No. Okay. So the first method was heat shock using divalent cation. Second is when you give the heat shock, like we use divalent cation, that's why it's heat shock plus divalent cation. The second method, which is not used mostly on bacterial cell, but is used on animal cell, which are like big cell or single celled um, um, eggs or zygotes, is called microinjection. It is straightforward as the name says. You take the plasmid in a small microinjection and you inject it inside the cell without killing the cell. So the damage has to be very like there will be some damage, but the cell will heal up because it won't be too much. It only will be the small poke that the injection will create, and these are tiny micro injections, and where you can directly inject the recombinant DNA into the nucleus of an animal cell. So you can write down micro injection. So no need to worry anything because you directly injected in the nucleus. So the job is done. And an animal cell, remember that animals cells are eukaryotic cells, so they have a cell membrane and then also have a nuclear membrane. So this is directly injecting recombinant DNA into the nucleus of the animal cell. Okay. And then the third method is mostly used for plant cell, and it's called the gene gun method. Again, it's a fancy name given to the method, which is technically called biolistics. Both these methods like hint towards using a gun. Basically, you use a small device which is filled with microparticles. Like you must have heard of nanoparticle term, but nanoparticle are too small. We don't need that. We can work with microparticles, and these microparticles are made of metals which are non-reactive inside a cell. So either it is gold or tungsten. Now you must be thinking, using gold to make microparticle, isn't it a very expensive method? But remember, we are using microparticles. These are even smaller than dust particles. Okay. So then we use these microparticles, coat it with our recombinant DNA. Okay. So on the surface of these microparticles, you have your, so if these are the microparticles, I mean, an enlarged view, and on the surface, it's coated with your DNA. In this case, it could be a plasmid, so they're circular DNA, and then you bombard these particles onto a cell. So basically, it's like a gun. You are firing at a cell. at high velocity with high velocity microparticles made up of gold and tungsten. And they will just, again, just like an injection, they will be like thousands of tiny injections poking the cell at the same time. And for a very brief period, we do it because we don't want to damage the cell so much that it dies. So we just do it. And some of them, of course, hundreds of them will enter inside. And then we stop it, allow the cell to heal, and then the cell will have the DNA that we wanted to send inside. Okay. So gene gun is clear. This is mostly used for plant cells because plant cells have a cell wall which is very rigid and slightly, I'll not say impossible, but it's difficult to do microinjection on plant cell than it is on animal cell. Basically, IVF, we do the same thing. You know, remember IVF, people? In vitro fertilization?
Yes, sir.
Yeah, we do microinjection only where we put the sperm into. So for plant cells, and they have also more tolerance, so we use gene gun. Okay, just write down in this method. This method is suitable for plant cells where this method is suitable for plant cells where high velocity microparticles of gold or tungsten and why we use these metals? Because they are non-reactive in a cell. You know, like magnesium, iron, all of these metals play a role in chemical reactions in metabolism inside a cell, right? They are cofactors for many enzymes, but gold and tungsten are not. You won't see gold as a activating or cofactor for any enzyme in metabolism. Of gold and tungsten coated with desired DNA or foreign DNA is bombarded on the cells. Okay, so that is gene gun or biolistics. And the fourth one, there is one more method which we have already talked about. And if you don't want to use heat shock, microinjection, or gene gun, then you can rely on some natural pathogen that actually can do this for you, like using agrobacterium or some virus which you have modified or disarmed. So we call this method as using disarmed pathogens as vectors. Pathogen vector. Okay, but you have to be sure here that the pathogen is disarmed, which means it won't cause any infection or disease inside the cell, but will also transfer our DNA that we want it to transfer. Okay, but since there's a chance of what if the pathogen, and we don't have to kill, we also have to make sure that the pathogen is not killed. So if you are disarming the pathogen using any technique, that technique should not kill the pathogen, but keep it in a form where it can still function as a vector without causing any pathogenicity. Okay, so for reasons, and this method is not widely in use now, especially to do science with human related therapeutics, we rely on making competent cells using heat shock, microinjection, or gene gun method. Any questions, anyone? I'll stop here if you have any question till now. So till now, I have told you all the methods and techniques that you needed to understand before you can address the final question, which is a 5-marker question. Often it comes in exams from this chapter. So these were all the principles and methods that you and technologies that you need to understand. So anyone has any doubts still here? I'll stop for a couple of minutes and take doubts if you have. Aisha, if no one has any doubt, just give me a thumbs up so that we can move ahead. Or you can take a minute and think. Go through your notes quickly and see if you have a question. Those who have no questions are either doing good job of revising regularly or are doing a bad job of utilizing these classes. Yeah. No questions? Okay, perfect. So let's move to the one and final question. We have studied till now all the principles of biotechnology. Now write down this question. What are the steps for synthesizing a recombinant DNA? And using it for getting final synthesized product. So basically from A to Z. What are the steps? This question can come in all different flavors. List the steps in specific sequences which are present in recombinant DNA technology, or in any other way. So basically what they're asking is, how will you make a recombinant DNA right from the beginning and use it to get the product for which you were making that recombinant DNA? Okay, make sense, everyone? Now let's see what we have studied. Okay, so what is the first thing that we will do that you people have also done in your practicals successfully? So first step you all have done. So you are, if I say there are 5 steps or 6 steps, you are already 15 to 20% biotechnologist right now. What do we need first thing to make a recombinant DNA?
Gene of interest.
Exactly. We need a gene of interest. And where I can get my gene of interest?
By isolating the DNA.
Yes. So first step is isolation of the genetic material, right? And when I isolate this, I have to isolate the whole genome, because I don't know, I cannot just isolate my gene from a cell without isolating the whole genome. So I have to isolate the genetic material itself. In this case, it is DNA. If we want to isolate RNA, that also we can do by RNA isolation techniques. So DNA isolation technique, you already know about, right? How do we do it? What do we need for this? So DNA is present in nuclear membrane. So we need enzymes which can burst open the cell wall, the cell membrane, and the nuclear membrane, and everything inside should come out, right? This is what we will do first, correct? This is what you did in your practicals also. Remember that? If you were using a fruit, you were just using a detergent maybe. So in schools, we can use detergent because that is one thing that burst the cells open and everything comes out. And then what you did? You specifically made DNA precipitate in chilled ethanol. Remember those steps? Have you done it or not? I think you have
Yes,
done
sir,
it,
we've
right?
done it.
Yeah, right. So you chilled ethanol, poured it from the side, and then you pulled out some stringy like thing, right? We have discussed this in chapter 6 already, where isolation of DNA. Yes or no?
Yes.
Yeah. So suppose we got the DNA. Next thing we need is we have to break it into smaller pieces. And before doing that, we have to get rid of all the impurities. So this you might not have done in your school. You just got a spool of threads together, and teacher might have told you that, see, this is the DNA. But that's not just DNA. That is DNA plus a lot of other things. But when we do scientific research or when we do biotechnological research, we need pure DNA. So anything that can be present with DNA, we need to degrade it. So we can do that by using enzymes. What are the enzymes that we can use to get rid of other thing? If it is protein content, we can use proteases, lipases for lipids, RNases to get rid of RNA, and amylases to get rid of.
Lysozyme.
Yeah, that is a different thing. I'll come back to that first. So this I will teach you now, but I'm talking about what you already know from the previous chapters, right? We can use proteases and get rid of all these things. Now we are only left with DNA, okay? That you know. But one problem sometimes is to open a cell itself. That is where what Iram says come into picture. Different kind of cells, a bacteria cell, a plant cell, animal cell, a fungal cell, which is yeast. When we work with different kind of organisms, we first have to break open the cell properly, because if that is not done properly, your yield of isolation will be very, very low. And if your yield is very low, from that low yield, getting your gene of interest is even more difficult. So we need a higher yield. And for that, we first have to break the cell properly. For that, we can use another set of enzymes, which helps us to go through the cell wall properly, or break the cell wall properly. Now, cell wall containing organisms are bacteria. And what is a bacterial cell wall made up of? Remember? Peptidoglycan. This you have all studied in junior classes. So we use an enzyme called lysozyme, which can very effectively burst open bacteria cell. And if we are working with plant cells, and we want our gene of interest is present in a plant cell, we know that the cell wall of plant is made up of
Cellulose.
cellulose. So what is the enzyme that we will use to break it open?
Cellulase.
Cellulase. Yeah. This is the same enzyme which is present in the gut of herbivorous animals, which are obligate herbivores in nature, and that's why they can digest every source of starch or cellulose from the plant. So when they eat leaf, humans cannot survive on leaf of any plant. We can survive on leaf of some specialized plants, but not in general grasses or tree leaves, because they contain most of the starch as cellulose, and that we cannot digest. We can only digest starch because we have amylase for that, but we do not have cellulase, so we cannot digest cellulose. Okay, but animals like cattle, cows, buffaloes, they all can do that. Okay, and if we are working with a fungal cell, which, for example, a yeast, if my model organism is a yeast from where I want to take out a gene of interest, then I will use the yeast cell wall is made up of
Chitin.
chitin, and we will use the enzyme chitinase to break open the cell. Okay. So all these are used to break open the cell and release DNA in the solution. After that, we will purify and we will get rid of other impurities using other enzymes that degrade protein, lipids, starch, etc., etc. Okay. Okay. So ultimately, when you have the precipitated DNA, so DNA ultimately precipitates out when you put chilled ethanol as fine threads, and these are the things that you need. Suppose now you have the genetic materials. So what will you do now? Very good. First step is clear to all of you. Just a repetition of what you have done till now. So you have the DNA now. Okay, everyone. Now we did not need the whole genome. What do we need from it? A specific gene that we want to put in a plasmid and send in a bacteria. So now we need to cut the DNA at specific location, which is called cutting the DNA using
Restriction and DNA.
yes, cutting the DNA using restriction enzymes. In our case, it's endonuclease, not exonuclease that we use. Now this we know how we will choose which restriction endonuclease to use. We will choose it on the basis of what segment of DNA we want to cut out. So we'll first go and check the sequence of our gene of interest. We will check upstream and downstream sequence, and then we will see the upstream and downstream sequence. Does it contain a cloning site or a recognition site for restriction endonuclease? And whichever restriction endonuclease can cut my gene of interest a little up and down, having some extra regions, I will use that, and I will treat my isolated whole genome or the whole DNA with that enzyme, and I will get small pieces out. And we also have already studied how to get our gene of interest specifically out from all those fragments. Remember? We will use a method called agarose gel electrophoresis. So once restriction enzymes have done their work, then we'll take that sample, run it on a gel, and then I will get my band, like the size of the DNA, which is my DNA of interest, or the size of the gene. I will get that equivalent size band on the gel, and this is how I can cut that DNA using this vector, get my gene of interest. Is that clear? So this is the second step, agarose, and this is the, you can say 2B or third step also, but it is the part of this step only. You have to do agarose gel electrophoresis to be sure that your enzyme has worked properly. I'm writing here endonuclease. And then we'll go to third step. We have our gene of interest now. What we'll do? In some cases, now we have to prepare a recombinant DNA, right? Third step logically is to prepare recombinant DNA. You have the smaller part, you have the plasmid, you want to insert the smaller part into the plasmid, correct? Does that make sense, everyone?
Yes, sir.
Yes.
Yeah. But sometimes before doing that, if your gene of interest yield is very low, then you would not like to go through again step 1 and step 2. Once you have your gene of interest, even you have very few copies of it, what you would like to do is you can amplify that. And that amplification is done through a technique called PCR. You've been taught this. What is PCR? So amplification of
chain reaction.
Yes. What does polymerase chain reaction mean? What do you understand from it? This is the third step. So what is PCR? We got a full form. Yeah, tell me.
We produce multiple copies of the gene of interest.
How do we do that? How do we produce multiple copies?
Through PCR.
Yeah, right. Through PCR, you're correct. But that cannot be the answer, no? The answer is how. You're telling me like the name. PCR could be anything. You just know it as polymerase chain reaction because this is what is taught. Could be police control room, which it actually is. PCR vans are police control room vans. What does the word polymerase chain reaction means to you? So can you see the figure that I have taken it from your NCRT only? Yeah. So let's say this is a piece of DNA that you are interested in amplifying and making more copies of. You have just, let's say, one copy to begin with. Okay, so it's a simple thing. How does replication happens in a cell? In a cell, a double-stranded DNA opens up and polymerase comes. Polymerase is the enzyme that comes and sits. And we then know we have nucleotides, right? Which keep on adding. So polymerase keeps adding nucleotides one after the other. It reads one strand and make a complementary strand, right? By adding nucleotides. Nucleotide, you are all familiar with these words. So what we do, first step is denaturation. We have to open our gene of interest. And for that, how can we achieve that denaturation? Very simple. DNA can be denatured by heat. If you apply heat, DNA opens up. Okay. So that's why we say, if you want to denature any kind of pathogen, for most of the pathogen, heat works. If you boil them, they're gone, they're dead. Even if their cell wall is not broken or burst, inside the cell, their DNA is all denatured, it's open, it has lost its configuration. So that pathogen, even if it goes inside your body, it won't do anything because it does not have the DNA anymore. But some bacteria can tolerate heat. Okay. And those bacteria are thermophilus bacteria, which we have isolated from very hot places like hot geysers and springs. Because when Earth was cooling down, there were regions of water bodies which were very, very hot on prehistoric Earth. And these were the places, small ponds and water bodies, very hot regions where life started evolving. And some bacteria have evolved to be called as extremophiles. Philic means loving. They love very extreme environments, so they can survive in a very high density of salt solution. So they are called halophiles. They can survive extreme temperatures. They are called thermophiles, and so on. Right. So from a thermophilic bacteria, scientists have isolated their DNA polymerase. So when we are doing this reaction, we have to add, for the next step, we have to add DNA polymerase. But if I put a normal DNA polymerase from an animal cell or from any normal organism, that DNA polymerase will also get inactive or denatured because that's a protein. Protein also, gets inactivated or gets degraded at high heat. And this heat is how high? it is around 94 degrees Celsius, very close to boiling water. This much heat we apply to our piece of DNA so that both the strands open up. Okay, and you can see in the next step, strand 1 and strand 2 both have opened up. You have added a heat tolerating polymerase, which will come and sit here very successfully because it can tolerate heat. And we got it from a bacteria called Thermus aquaticus, TAQ. So the term TAQ, which is called Taq polymerase, it stands for T for thermus, heat, AQ for aquaticus, which is this is the genus and species. So this is the bacteria's name, Thermus aquaticus. From this bacteria, we got this DNA polymerase. So it's called Taq polymerase. So you have to add two things, a polymerase and some synthetically produced deoxynucleotides. If you are making RNA, you will add a different polymerase and add ribonucleotide. This is deoxy because we all want to make DNA. So deoxynucleotides plus Taq polymerase will give you this process is called annealing. But for that, you have to give them primers. Primers are nothing but a small sequence of around 10 to 20 nucleotides which you have made, which will go and bind at your gene of interest and will help the process. Okay, so you don't have to start from zero or first. You don't have to add the first polymerase. You can start with the primer and then continue adding after the primer. Now these primers comes in, you have to use two primers as you have understood by now. One which will act here, primer A, and one which will act here because there are two strands that opened up, right? And you would like to synthesize your DNA on both the strands, that then only you can multiply it. This is basically following the rule of semi-conservative DNA replication. So one primer is called forward primer and the other is called reverse primer. Basically, it's a sequence that goes in forward and reverse orientation. So 3' to 5' and 5' to 3'. But you know that polymerase only works from 5' to 3'. So in this case, it will work this way. In this case, it will work this way. So this step is called, this is first step. This is second step called annealing. So you are annealing means pasting. So you are pasting your primers on the template DNA and you are extending it. So for extending, you will allow a lower temperature. So annealing happens. For annealing also, you have to lower down. Okay. And for extension, you will come down around 60 degrees or even lesser. So starting from 94, we come to somewhere around 72 degrees, and then we even go till around 50 degrees Celsius. And then extension, basically the polymerase does its work. And we started with 1, and after the end of 1 cycle, we have 2 DNA. Now in the second cycle, from 2, you will have 4. In the third cycle, you will have 8. In the fourth cycle, you will have 16. And it will grow exponentially, right? It just needs 30 cycles to make almost 1 billion DNAs. Just 30 cycles. Because every time you are doubling it up. You understand? Yeah. You understand, everyone?
Yes, sir.
Perfect. So 30 cycles, we get 1 billion pieces of our gene of interest, and we can store some and use the rest to make the recombinant DNA now. Okay. So this is third step, and then we go on to fourth step. Sometimes we don't do the third step where we already have a very good yield. So don't need to. But if you have, if you don't want to isolate every time, which is a tedious process, once you have isolated, we never again isolate. We just keep amplifying our gene of interest by doing PCR because that is a better way to do it. Okay. And then we'll go to the next step, which is inserting this piece, this one, these here, into a host through a plasmid. So fourth is insertion. Insertion of recombinant DNA into the host. And this we just studied in the last class. How we'll do that? For that, we have to make the host competent. And I taught you there are 4 ways to make the host competent. We can use, depending on which cell we are using, to as our host cell, we will make them competent. And because our plasmid will also contain a marker gene, selectable marker gene, we can also identify which of the cells have been successfully transformed and which have not been. So this point is clear, insertion of recombinant DNA?
Yes.
Yeah. And then we'll go to obtaining or synthesizing the product. Foreign gene product. And what is the product basically for every gene? Protein, right? So we're doing all of it to get protein. Make sense?
Yes.
Okay. Now, how do we do this? So because our gene, the DNA that we sent was a recombinant DNA, when it will express into a protein, because it is expressing into a host where it naturally should not have been expressed, right? It is a foreign DNA that we put inside a bacteria. So bacteria naturally does not produce that protein because naturally it does not have that gene. We put it inside using a plasmid. So therefore, this protein is called recombinant protein. You understand? There is a 1 marker question. What is a recombinant protein? You can write down. And by the way, I forgot to tell you one thing. This process here of doing a PCR, we also call it gene cloning by through PCR. Because what we are doing here is making multiple clones, identical copies of our gene proteins. Make sense? So this is a synthetic way of doing gene protein. Bacterial cell, and it will make copies, which anyways it will do. But before that, we also made copies, so it's called cloning through PCR. So when we put our cloned gene into a heterologous host, so write down, when any protein encoding gene, when any protein encoding gene when a protein encoding gene is expressed in a heterologous, and why we call it heterologous? Because hetero means different. In a different host or a heterologous host, it is called a recombinant protein. It is called a recombinant protein. Yes, Parker, because it's different. Perfect. So there, what will happen? The bacteria will culture, or any cells, we will culture them into big bioreactors, and we will make liters and liters, 100 to 1,000 liters of these bacteria. And later on, you can just purify your protein from these bioreactors, these big cultures of bacteria, and you got the final product. Okay. So how do we culture this? Write down. So we use, do you all have NCERT with you? I think that will be an easier way. Can you come to, I think the last figure of this chapter is a bioreactor. They show a bioreactor. Can you come to that figure, everyone? I will also try to pull it up. Yes, I got it. This image. Can you all see this image?
Yes, sir.
Yeah, let me make it bigger. Yes. Yeah, so what you see here is a bioreactor. It's a diagram of bioreactor. Oh, which page? Parker, which? I think it's in the new books. It's figure 9.7 on page 174.
No, sir.
Yeah. What is
204.
204? Yes, on 204. I guess then it's figure 11.7. Is it?
Yes, sir.
Yeah. 204. Everyone on this figure? Perfect. So this is where we make big volume cultures, 100 to 1000 liters cultures of the cell, host cell. Okay. So this is a simple bioreactor, and this is a more little advanced bioreactor, B. So in case A, what you see here is there is a motor. So this is a big vessel, and there is a place to send air, sterile air from. Sterile air means the air should not be contaminated itself. So the air has to be sterile. You are culturing bacteria, you would not like bacteria coming from the air to contaminate your culture. This is where making this bioreactor is a part of bioprocess engineering. Remember that? So when you do bioprocess engineering, you make a reactor. And what you do inside the reactor is genetic engineering. You have done genetic engineering already, you are just culturing it. So there is a motor connected to flat blades, which basically are used to generate broth, or to keep moving the culture broth. Why you need to keep it moving? So that the air that you are pumping from the down mixes properly. Every bacteria gets proper oxygen, so it grows properly. Nutrition is there, which is mixed properly. You have an acid base control. You can control how much acidity or basicity you want in your system. And you have steam for sterilization. This is the inlet for steam. Before you start an experiment, you have to send a lot of steam, very hot steam inside, to kill anything which is remnant. So you use the reactor to culture one type of bacteria, then you can wash it up, send in steam, sterilize again, and then you can culture a different kind of bacteria also in the same reactor for the next project. Okay. So it's very simple. You just need very thick walled big chamber with a motor, with blades, impellers, where you can mix the culture broth, an inlet for sterile air, a regulator to control acidity, basicity, pH, temperature, humidity, pressure, everything we control basically, which they have not shown. So these can be the things to control everything. Then we have steam inlet, and that's done. It's sealed, super sealed. In some other bioreactors, which are called sparged stir tank bioreactors, basically you send in, again, here you are mixing it, right? And you have these bubbles. So these bubbles are, you create bubbles inside the reactor from the gas, right? And what do these bubbles do? Bubbles increase the surface area through which oxygen can be transferred to each and every bacteria. Okay, so bubbles are always a good way to increase surface area. And bubbles basically means can come through frothing. So this is just called sparge stir tank. All the concept is same. These are also super sealed. They have just shown it like this. They are not open tanks. They're super sealed, sterile. The thing is here, the idea is to produce more and more bubbles, which will keep mixing along with the bacterial cells and will allow more surface area. So if you are growing a bacteria that requires, or if you're growing a cell that requires more oxygen, you can use the sparge stir tank bioreactor. In other cases, a simple bioreactor also works pretty well, not a problem. Make sense, everyone? So
Yes,
this is
sir.
how we'll obtain the gene product. Everything is happening on its own. Inside these tanks, the bacteria is replicating your plasmid, transcribing your plasmid, and translating your plasmid into a protein. So your gene of interest is also getting made into a protein. It's all biological. And the last thing that we will need to do is what? Procure that protein. So that step is called the downstreaming processing. Once your culture is done, it's grown properly, then you need downstreaming processing. And this includes, if it is a, after your biosynthetic stage is done. So this stage, obtaining the foreign gene product, is called biosynthesis stage. Why biosynthesis? Because synthesis is happening a lot of things. We are allowing them an environment where we can culture them. Okay. So after the biosynthesis stage is done, in downstream processing, what we do is we separate. So first step is separation. You separate your cells from the liquid in which you are culturing it, followed by purification. Okay, which are basically together called downstreaming process. So separation, you separate all the cells, burst it open, and then you purify the protein which you want. Once you have purified, the third thing which comes in downstreaming processing is suitable preservation. Because you have made so much of protein, whatever product it is, if it is insulin, if it is some other enzyme, amylase or any other enzyme, keratin, biotin, whatever you wanted to make, for any drug, for any disease, or for any medicine, whatever you wanted to make, you have made it in bulk, a lot of it. So you have to preserve it. It's not like you can just purify and use it directly. So to preserve, we need a formulation, right? So after suitable preservation, or I should say, for suitable preservation, you need to make a formulation. By formulation, I mean, how are you going to keep it? For example, your vaccines, COVID vaccine, you must have heard of news where people took COVID vaccine shots and they got side effects or reactions in their body, right? Did you see that? Hear that? People, are you there?
Yes, sir.
Yeah. So most of the time, those reactions were not because of the mRNA that we are sending in, okay? Because that will, of course, elicit immune response, but it will cause some reactions which you will not see in every person. Fever will happen in every person who will take vaccine, and some form of mild muscle pain. But apart from that, if you get other very severe complications, that might be because of the formulation in which that vaccine is dissolved. So that liquid that is injected inside your body is not just the the total or the pure RNA, it has to be preserved or kept in a formulation. That formulation is also chemicals. So we have to make proper formulation for clinical trials. Okay. Whatever drug we are making, it has to go through clinical trials, different phases, 1, 2, 3. I think third is where the human trials are done. Before that, there are animal trials. And if it's a drug, then you need to go through clinical trials. And to go through clinical trials, you have to preserve it in a formulation. Okay. Finally, if everything goes well, you do the control testing. Also known as the quality control testing. So, sorry. And then if it's passes, all the tick marks are there, then your drug is ready to come to the market. Okay. So these are the 6 steps. Yes. Isolation of genetic material, cutting that DNA using restriction enzyme to get your desired gene fragment, amplifying your gene fragment using a PCR. Once you have amplified that, insert it into a plasmid to make recombinant DNA, and then insert that recombinant DNA into the host, and then let that host cell grow in big cultures to get the final gene product, which is protein. And once that has been done, then you separate it, purify it, take out the protein, make a proper formulation in which that protein can be active and preserved, and then go through quality control testing, and it's done. Any doubts here, people? No doubts? So with this, we come to the end of this chapter, which is your principles and processes. And I would like to connect, would like to stop here for a couple of minutes and ask if you want to, before going into applications, is any part of this chapter you missed or was weak, or you want me to go over again? Just let me know. Or you have any questions? We can also go with questions. In the meanwhile, I will just start next page. No doubts, people? Yes, go ahead.
So when we obtain the desired gene, the gene of interest, we attach that to the vector plasmid, right? So the
Right.
vector plasmid is taken from the host organism?
Not necessarily, because we use bacteria like E. coli that do not that might not have the same plasmid that we are using. So we don't have to get it from host organism, but it should be compatible in the host organism. That's a good question. So I can only put plasmid in organisms where plasmids are stable and compatible. If I put a plasmid in an animal cell, animal cells do not have any extra chromosomal DNA lying around, right? If anything like that, if any DNA, extra DNA is lying in an animal cell around, it will be cleaved, not just by exonucleases, but by endonucleases also. It will be cleaved. So that's why animal cells do not have plasmids. The only extra chromosomal DNA in animal cell is present in mitochondria. But in bacteria, plasmids are stable, they are functional. So if we are using bacteria as a host cell, I can put plasmid in. Okay, and that plasmid need not to be coming from the same bacteria. It could be, for that matter, it could be a totally synthetic plasmid that came from nowhere. We created it in the lab with multiple cassettes of gene insertions possible. But since and because plasmids in general can be stable in bacteria, we can make that synthetic plasmid also work. For other kind of organisms as host cells like fungi, plant, and animal, we sometimes send the piece of DNA only with origin of replication directly, like through gene gun, and because we are putting it in the nucleus, right? Because in animal cell, nucleus is the only place where replication. It does not happen outside that region. Make sense?
Yes.
Yeah. But in cases, you're right. In cases where we are using plasmids, they have to be host compatible, not necessarily coming from the same host all the time. Yeah.
Yes.
Okay. Perfect. So let me share the. I've shared the next notebook. Can you see the notebook I've shared?
Yes, sir.
Yeah. So thankfully, if we talk about applications of biotechnology, there are many, lot more than you can study. In a even in a year, if you just, so people who take biotechnology as a proper course, if you do a BSc or a BTech in biotechnology, you study the applications and details of it for 3 to 4 to 5 years. So it will take that much time if we studied biotechnology in detail. But thankfully, here we are going to focus on two major things, two major aspects, two important things for which biotechnology is very, very helpful. So when we talk about applications, the two sectors that has been revolutionized by this technology is one, agriculture. And you can understand why, because it provides you with food, right? And second, so food shortage should not be there. You cannot sustain food shortage. We need lots and lots of food. We need nutritious food to feed the ever growing population. The second is medicine, right? Because this is health, right? So health and food, these two sectors are very important, and these two sectors are also hugely impacted and changed by biotechnological tools to a great extent. Okay. So, but before, so we will study these two, the application of biotechnology in agriculture and in medicine. Apart from that, in this chapter, we are also going to learn about how do we make transgenic animals. What are transgenic animals? Wait 1 second. Transgenic animals. And we will also talk about the ethical issues that are associated with doing everything that I have talked till now and that I will talk after this class. So transgenic simply means, the word trans means it comes from, some other side like cross, and genic means gene. So if I put a human gene in a yeast, the yeast becomes a transgenic yeast. If I put a gene from a bacteria into a fruit fly, that fruit fly becomes a transgenic fruit fly. Make sense, everyone? Yeah. Transformed by biotechnology. Write a question, which is a very basic question that sometimes is asked. What are the main what are the most critical research areas of biotechnology? What are the most critical or most vital or important research areas of biotechnology? The first is everyone who is a biotechnologist or who goes into the field constantly works on these three areas. So there are three critical research areas. Basically, these areas never sleep because they always try to find something better and better. First is we will always keep needing improved catalysts, right? So first is providing or searching for the best catalyst. It is not just chemical catalyst, but also biological catalyst. And these catalysts come in the form of improved organisms, which means improved bacterial cells, modified bacterial cells, or when I say organisms, basically I talk about microbe, bacteria, yeast, etc., or a pure enzyme. So these are the two things that you are actively developing in biotechnology. You always need better and better microbes that you can culture, or better pure enzymes that you can get out of your cultures, which means getting better products simply. Second is, even if we have the best catalyst, which is a product of genetic engineering, we need best conditions in which this works. So second is doing the best bioprocess engineering, which is creating optimal conditions. Agree? So 30 years ago, the kind of reactors we used to have, or the kind of incubators, refrigerators, the kind of precision air conditioners we used to have to do biotechnology, 30 years down the line now, we use much more advanced versions. So creating optimal conditions through engineering. And this includes all sorts of engineering, chemical, mechanical, electrical, software, everything. Through engineering for the catalyst. So this is the same catalyst that we have talked here, for the catalyst to work. Okay, this is second thing. And third is, if you do these two things constantly good, then the third thing you have to keep doing research in is the area of technologies that are required for better and better downstream processing to purify the protein, right? So better downstreaming or downstream processing, I should say, better downstream processing technologies to purify the protein. If we have put a gene in, we will purify a protein, or in sometimes we also make just organic compounds, just a drug, not a protein. Okay, so these are the three areas which never sleep. We always have to keep doing better and better at this, because this is on which the whole application field is based on. Everyone's clear? Any doubts in this? Perfect. Now, let's go to the first bigger picture, which is biotech applications in agriculture. Okay. Now we are not going to cover this in today's class, but I'm going to give you some homework because I think we don't have much time. First, I'll ask you a question, okay? And then I'll tell you what you have to go and read on yourself before you come to the next class. And two things we all have to discuss. So we have Aisha, Iram, and Rabia here. From next class onward, because we are moving towards the end and we have to finish the syllabus, and also you people will start doing your revision tests, you will start solving sample papers on your own at home. So to incorporate for your doubts from the things, sections that you have missed, so we also have to cover that. So from next class, your classes will be 2 hour long. It'll start at the same time, but go for half an hour extra in every class. Okay? Everyone is fine with that, so that we can finish remaining things, including what we have left behind, and we get some time for doubt solving and discussions also. Aisha, Iram, Rabia, just need your confirmation as a thumbs up.
Yes, sir.
Okay, perfect. Yeah, so just wanted to inform, if anyone has any issues, just list them down on the WhatsApp group. Inform us about your issues and we'll see what can be done. If there are no issues, then from next class onwards, Wednesday onwards, including Wednesday, we will have a 2 hour long class. Okay. Now for Wednesday, I'm aiming at going through the major part of applications of biotechnology. So what you have to do is you have to learn, not learn, just read about two things. Remember, we talked about tissue culture when, not in this, but it's a asexual mode of reproduction that we can do for many plants. Remember tissue culture? Plant tissue culture, the topic. So write down these topics that you have to prepare before you come in next class. And these are all from your previous classes, of course. So tissue culture is first. Just focus especially on plant tissue culture. Then the concept of totipotency. Totipotency and multipotency. What are these two concepts? Okay. You know about meristematic tissues, right, in plants that you have done in class 11th. Meristems. So just read meristems also. And yeah, I guess that's all. Okay, cool. I think that's all you need to prepare. Anything I'm missing? No, GMO I will teach you in the next class. Yeah, so these three. This is your homework. Just write it down. Written?
Yes.
Perfect. So in the next class, I will focus on, we'll start with Green Revolution. If you want to read, it's up to you. You can go and read from your book so that you are better off in the class and you can discuss more. And we'll talk about BT cotton, one of the important topics that is important both from boards and entrance perspective. Okay, next class. But this is important.